would like to thank Indian Academy of Sciences for giving me a chance to present my work with this august audience. And today I'll be speaking on brain sex differentiation in fishes. I put on quote because the idea for brain sex differentiation in fishes mostly came from our laboratory. So we were trying to prove that the brain also play a role in the process of sex differentiation. Usually gonads play a role. So we have proposed an hypothesis, and most of our findings, maybe end of my lectures, maybe you may get convinced partially or fully that there is a possibility for brain sex differentiation. First of all, what is brain sex differentiation? Most animals, the gonads dictate brain, and based on that, the sex hormones coming from the gonad have an influence on brain, so that we can have a situation of a male and female brain. And when you talk of brain sex differences, naturally you will see whether it's gender specific. So certainly it is gender specific, because most of the times we always feel that, multitasking, but men cannot do multitasking. If you see men, they have isolated one task, they'll be thinking on that, and they won't do beyond that. I'm very sure actually. Most of you know that. How many of you know that you have called your son or daughter about on the exam day? Most of the time I fail. My wife teases me, asking me, today did you ask your son? I said yes, the exam is yesterday. Okay. So that's mostly the trend, because we are not very much into multitasking, but women can do extraordinary multitasking, because the white matter in the females are more. We have a gray matter, so we are naturally on one focus. And then men also have a different problems. We have an amygdala. Because of that, we are more aggressive. So either we fight on a situation, or we will escape from that fight. Flight, we will flight, that's all. But we don't have the intermittent thing. But the females have this intermittent also. They will fight, they will flight also, they will join with that also. So we never forget. Sometimes we fight means we never forget. The female, sometimes they will forget it and they will get along. You can see, for example, in olden days, even nowadays also, we wait for water. They will line in queue, and after that, what will happen? Once that during the queue, they will be very strict. After that, they may share food also in the back home. So that way, also the hormonal response behavior is somewhat different. And also, so that's what I'm saying, that a woman can do many things at one point, but men will always get puzzled whenever, many things are coming in hand. Especially when you become old, you can see grandma will dictate grandfather actually. So always it happens actually. So even in my case, my grandpa is MA English literature, my grandma is only 3rd standard, but she will dictate him what to do. Okay, I have seen on my own eyes. So I agree with that. Maybe when I was very young, I could not understand what is happening, but now I know this is happening because of the differences Almighty has given us. And then also brain also participate in several of these monoamines, give different kinds of inputs, and especially for men and women, they don't have a similar parameters. Even we have like right left also, you can see there are a lot of differences always happen in the human as well as mammalian brain. And you can see number of these hormones behave differently in men and women. You can see, for example, if you take prolactin, I have not mentioned here, prolactin is a classical example. Prolactin is a hormone which is lactogenic. You can see the differences in prolactin hormone when female is not pregnant, and then female when pregnant, then the levels of hormone goes high. Instantly, if you see the single father who did not get married or continue to live a single father, they have quite a bit high level of prolactin, with the normal woman don't have actually. So it will be in between the lactating woman versus normal woman. Prolactin levels are very high in single father. So there are differences in hormone based on the behavior also, and also based on the sex also. And then you can see some of these peptides which are coming from the posterior pituitary, especially oxytocin and vasopressin. These are 9 amino acid nanopeptides. They behave differently also. For example, if we take the Perera-Oles, they bond for life actually. So they will be husband-wife forever. And if they get separated, the male will have a solitary, similarly female. But whereas the same kind of Montagne, they don't behave like that actually. So there are plenty of examples coming in the mammalian literature as well as human that there is a perfect brain sex dimorphism. And also the feedback regulation, if you see the female has got both positive and negative feedback, which is not there in the men. Men always have one negative feedback, that's only negative. And because of that, the female is most advanced or evolved organism in terms of evolution actually. So because of they have the cyclic center in the brain, because of the female is again evolved. But when we have all these literature, so we thought why not in fish will have such kind of a brain sex dimorphism. That was our question actually. So we started this journey maybe 15, 16 years before, that there should be some differences because we see some examples. I can show some examples why we believed that. So on the other hand, like a negative and positive, fish do not have a positive feedback. They're all negative actually. Whether you take male or female, there's only negative feedback. This was a big disappointment for us that to go about it actually, because there is a positive negative for the mammals, but we don't have. But still we on fish to understand whether there is any kind of gender difference we are seeing actually. So that was our question. And then this question is a little bit important because some of the studies shows that we can change fish from male to female. Changing fish means I am saying that changing the sex of the fish functionally. For example, if the male became female, they will start to produce ovary, eggs, everything will come there. Similarly, if the female becomes male, they will also become producing sperm. So it's a fully functional sex reversal. The fishes are the only vertebrates which are called champions of sex reversal. When you come to frog, only one way is possible, female will become male. And in reptiles, temperature can change. But once becoming a sex is established, changing can be done only in fish. And we can either do naturally or we can do with the hormones. And then gonadal transdifferentiation always happens first than the CNS system actually. So in this case, always gonad will change first and then brain will follow that actually. And these are some of the examples which we see, Protandrus and Protogynous. This fish lives as a male for 1 year, then second and third year they will become female because the male is bulky. So naturally the male is get harvested, then the biggest male will become female. Similarly here also if you see the female will get harvested, and then because of that one of the female will become male, and then the school is continued actually. So this is a natural way of doing it. We can also stimulate this by treating hormones early times. So if you treat with estradiol, you can have an XY female. Similarly, if you treat with XX, treat with androgens like methyl testosterone, then you can have an XX male. So the alphabet of chromosome is just an alphabet. So we don't call it as a significant. So there is genetic sex, hormonal sex, environmental sex, plenty of these are there. And then, so this is what I was telling, that you can change it at the early stage, you can change it to the either sex, whatever you want. And then here also the gonadal differentiation takes place first compared to brain. And there are some other fishes which is what giving us an example that certainly brain sex differences there. These are serial sex changes. What happens if you have a pool of fish and then one male will be there and 3 females will be there. So the male will do the parental care. And if you remove the male, the biggest female will become a male actually. Then if you bring back that male again to that area, or if you taken in an arbitrary lab setup, then if you take that male and then bring back that male, the biggest female which changed sex will revert back. So these are called serial sex changes. All these happen in 1 week time. Okay, so here you can see the it will bring the female to egg lay and then it will shed the male and then it will protect the young ones. So you can see that behavior from the very beginning. So this gives us some kind of an interesting idea that certainly brain will show some response. So it is always showing that kind of a behavior means there is some response will come brain. So we thought that it's worth investigating. So these are some of the fishes which we essential models, which is coral reef fish like our the clownfish. They live in a small school. So then that one male will be 3 females. So imagine if one more male comes, then the male has to fight out which male will be there, like a lion's pride. So this is one example. Another is Thalassemia duprae, which is again a coral reef Hawaiian fish, a little bigger than the one which we saw in Thalassemia duprae. And then there is also other fish models. But unfortunately, I didn't have a chance to work on these models because you have to scuba dive and catch fish. I never even swim also, so it's very difficult for me to scuba dive. But somebody told me that swimming is not needed, you can scuba dive. Still, I didn't have the courage to learn that. So I thought, why not we work on fish models which are available with the genetic sex populations. We have a Nile tilapia. We started our work way back in Japan with XXXY populations. We know that it's become XX and XY. So it was a very good model. And then later on in India, I started working on these fishes, which are domestically available for me, and also which is a very important fish in terms of food fish in India. So most of my work first started with Gary pinus, which is an African catfish, which was not accepted to research by DBT, but SERB always, DST always support that. But later on, we switched to Batrachus, which is an Indian catfish. So we rear this fish and also have collaboration with that one. So starting with tilapia. Tilapia is a very good model because it's a fortnight breeder. So we have an XXXY population. So we thought to work with this first, and also I have a chance to have the success of XXXY population. So we started this work. And the life cycle of Gary pinus is 1 year actually. So you have to wait for a year to do this work, and we don't have any XXXY population. Batrachus also similar actually. So first we decide how to proceed this topic actually. So we thought first to go with the GNRH, and before that we also worked on some of the amines we also worked. So I will come for the GNRH and then go back for the amines actually. So GNRH is an important releasing hormone set right the gonadotrophic axis. So we started analyzing the GNRH protein, both XX, XY population. You can see this XY male, it appears very early, somewhere around 7 days, and disappears and then comes back again. On the other hand, in the female, you can see the appears around 15 days once the gonadal differentiation happens, and then it gets stabilized. So there is subtle difference actually. So it means that there is a GNRH coming up and then goes off and then comes again. But on the other hand, female, it comes off little later after everything is established. So one area we thought there is some differences, and then we thought there is also something in gonadotrophins, but we didn't get any changes in gonadotrophin. So GNRH difference, dimorphic difference in terms of appearance was first clue. Then we thought to go for amines, and amines is very important because they set right the GNRH axis, especially serotonergic and calcitonin system. And we got some information before that if you block serotonin, which mimics like estradiol, especially early time. So this was a kind of a causative response. You block serotonin in brain, and then it happens in terms of gonad also. So this was a small study available before we started. So we thought to clone this rate tripling enzymes, that's tryptophan hydroxylase, and see what happens to this enzyme, because certainly there should be a difference between male and female. So we cloned this, and then we found that the early expression XX and XY only have this particular enzymes. In 0 day, you can see some expression, but because we use the old larva or old fry. But fry day onwards, we have checked only the brain region, and you can see the females does not have, but the male do have extraordinary amount of TPH with the quantitative PCR in terms of RNA. And then we want to compare this ovary and testis also, because they express little bit serotonin. So then we found that it is not showing any dimorphism, only brain dimorphism is seen. Then we wanted to localize this in the transcripts. We localized, again, nothing is there in the male, but nothing in there in the female, but male have got plenty of transcripts of tryptophan hydroxylase, which is the serotonin synthesizing enzymes. Then we also see the protein. Again, the XY male have got a specific expression of protein is found in the XY male, but not in the XX female actually. So this gives the idea that clear-cut sexual dimorphism. Then after 30 days, this does not happen. Then we went on to Claris batrachus or Claris Gary pinus also we checked, and then we found that since we don't have an XXXY population, but then you can see around 50 and 75 where there is a differentiation takes place. Again, the overexpression is seen only in male, and then we also see the protein levels especially in the preoptic area. Everywhere the male shows a very high expression actually. So which tells you that the TPH is selectively found in male during early time, but not in female actually. Then we also analyzed the precursors and the metabolites which are coming from the serotonin. Again, serotonin TPH. Then you can see the serotonin levels and 5-HTP, everything is going high in the male. So the enzyme and the products, everything are at higher levels in the males. And then we also reversed by changing sex. So we did an MT treatment. MT means methyl testosterone. We also treated with estradiol. instead of estradiol, we put PCP here because it mimics like estradiol. You can see that the PCPA, whenever you give PCPA, that means estradiol level goes down. So that tells you that the tryptophan hydroxylase is towards male but not towards female. That means there is a selective elevation of serotonin in the males but not in the females. So that explains me that the GnRH in the early time, because of this serotonin, it appears and then goes off and again stabilizes. So this was the first finding which we did, and we are first to show the brain sex dimorphism in terms of serotonin and the enzyme, and which is a kind of a causative response, not consequence. But all other studies later on we could get only consequence. And then in the females, we started working on the candidate gene brain aromatase, because females have got a lot of aromatase activity overall in the brain. And then we also could see the expression of both the aromatase always found high in the female but not in the male. And then you can also see the expression is pretty high, and also the transcript which promotes the aromatase enzyme also very high in the females actually. So then we also checked the catecholaminergic system, which is another system which is operating in GnRH axis. So you can see all of them, especially the enzyme tyrosine hydroxylase, which is a rate limiting enzyme for catecholaminergic synthesis. All of them shows very high expression in the females but not in the males again. So that tells you that the serotonergic system is there for the males, whereas the catecholaminergic system is there for the females. But once the sex is established, it becomes normal actually. So the difference only available during the early time when there is a differentiation takes place. And then we also measured the metabolites and also the products that are coming from that. Everything is showing very high levels. And then you can see the female always tend to show, and then when you reverse with the estradiol, you can see the estradiol promotes. That means female means you have a lot of catecholamines. It promotes. When you give the methyl testosterone, it doesn't promote actually. So then we have we also studied the GDNF family, which is also a dopaminergic system establishing. That also is towards female, very high. And then NPY, which is another peptide which is stimulating GnRH, also very high in the females but not in the males actually during early development. So number of markers we have listed, and then we have found out that most of them for the females, but only serotonergic and dopaminergic system, this is TPH is for the males actually. And then we also tried to put these sex steroids reversibly, like putting estrogen in the male and the testosterone in the female, and we have used
The pumps are self-powered through the reliable process of osmotic displacement. Here's how it works. Alizet osmotic pumps consist of 3 concentric layers: the inner drug reservoir, the osmotic layer, and the outer rate-controlling semi-permeable membrane. The reservoir within the core of the pump is filled with the test solution to be delivered. When the pump is implanted, water is absorbed through the outer membrane. The water entering the pump expands the osmotic layer, compressing the flexible impermeable reservoir. This enables the test solution to be released through the exit port at a controlled, predetermined rate.
So we used the osmotic pump to release estradiol and testosterone selectively, and then we also found the changes, whatever transcripts we have shown, that especially it is towards female and male. You can see the changes that is happening when you mismatch it. So that also again confirms that some of these markers which we identified are showing sexual dimorphism. And then we also showed at the level of brain with the immunohistochemistry, whatever we have found out that mismatch because of these hormones, we can see the changes, whatever markers we have described. So overall, we have found several markers for female side, but for the male side, we could always get only the serotonergic system at early development, which is showing a cause response. Remaining all of them is a consequence response, re-establishing that the gonadal will dictate the brain in terms of brain sex differentiation actually. So we also established a neurodegeneration model with this kind of an idea. And also we are now working on creating neurodegenerative models, especially using annual breeders. Usually you have daily breeders like zebrafish is a good model, but we are now establishing annual breeder models which are working very well for neurodegeneration. So all my studies over the time now helps me to understand this behavior pattern, especially neurodegeneration, which is different between male and female actually. So these are some of the publications and my research team, which are presently working with me. And then most of this work was done by the postdoc, with my Mamta, and then my wife Sudha also did, along with late Professor Aparna Datta. She was with her actually as a PDF. And I'm thanking late Professor Aparna Datta for a lot of intellectual exchanges between us, made me to grow on this topic actually. Thank you very much.
I would have a big question. Do you observe any morphological differences between male and female?
Yes, sir. But in catfish, we don't see. Tilapia, we do morphological differences. The male is little more heavily pigmented, but the female is little light.
But when structurals take part,
Yes, sir.
morphology
Yes. Because they show that aggressive pattern, and which will be very much seen in this coral reef fish actually. But this kind of domesticated foot fish shows very less changes. But this, the ornamental fishes shows fantastic changes sort of completely. You can even, while changing itself, you can say that this is going to become actually. So you can see that. And then interestingly, if you put them in a two different glass, people always say that the fish have got less eyesight, but still in the glass itself, if the male is there, it won't change sex. So that was very interesting observation. One of my Japanese colleague worked on that, but he did not continue. But he showed me that put the Trimacanave male in a glass with the same glass. When that male is there, it won't change. But people always say the eyesight is poor. It is not in the next aquarium, but still it didn't change.
any other group of animals do you find this phenomena?
Only marine fishes have this phenomenon. So if you want to work in a freshwater fish, then you have to simulate, like by hormones. You can only do. Natural behavior is very less in freshwater fishes actually.
What is the typical sample size that you are using? How many, when you are doing the experiments, how many fish do you?
Usually we used to more than 5 or 10 actually. So because we used to put all the fish, for example, estradiol, we used to have 100 fish. So when you put estradiol to them, so almost like a 95% will reverse actually in estradiol, especially in catfish. But in the case of testosterone, interestingly, testosterone is less effective than estrogen, at least in catfish, unlike other fishes, where again we will use around 100. But when we do for sampling, always 5 to 6 for each of the experiment actually.
For this development, is there any stage which is, because it's a transition, you said yes or no, so it just transform, but is there any stage which is in between?
It's a very good question, because some of the fish will be requiring the change can happen only early. Some of them will change early and juvenile, some of them in adult. So the work model which I am using, like tilapia or catfish, can only change in the early development. So it does not show any. That's why when I mismatch the steroids, the molecules will show change, but it cannot become a female or male. Only thing is they will have a disturbance in terms of fertility that may have, but they don't change sex once they become adult. They are very dominant when they become adult. But when you are young, you can change. But certain fishes, like I told, coral reef fishes, even adult is succumb to change. So each fish species have got a different window, but on each window they can change completely.
The XY male in the on the Y chromosome, there must be genes that are expressing. When the XY male start behaving like a female, what happens to those genes on the X chromosome? Do they stop expressing on the Y chromosome? Do they stop expressing?
So the answer is available with Medaka. We found a DMY triplicate of the DMRT1. It has been established as a sex-determining gene. Later on, AMH also found. So whenever you treat with this estradiol to those animals, the aromatase expression shoots up. Though aromatase in autosome, it shoots up and it down-regulates the DMY expression. So that's why the sex-determining gene Y chromosome can be easily, regulated down with the hormones.
Does that happen in all cells or only in gonadal cells?
Only in gonadal cells. But instantly, this kind of work is available only with the few fish species where sex determining gene have been identified. For example, zebrafish, there is no sex determining gene till date. So the answer is we don't know how it is. But certainly XY is a mere alphabet for them.